started. Hello, everybody. It's my pleasure to introduce today's speaker, who is Dr. Christos Davatzikos. He's the Wallace Miller Professor of Radiology at the University of Pennsylvania and Director of the Center of Biomedical Imaging, Computing, and Analytics. He's also an IEEE Fellow and a Fellow of the American Institute for Medical and Biomedical Engineering. Dr. Davatzikos holds a PhD in Electrical and Computer Engineering from Johns Hopkins University. And after that, he joins the faculty there in the Radiology and Computer Science Department, where he founded and directed the Neuroimaging Laboratory. In 2002, he moved to University in Pennsylvania, where he also founded and currently directs the section on biomedical image analysis. His interest is mostly in medical image analysis. He oversees diverse research programs, ranging from basic problems of image pattern analysis to machine learning, in a variety of clinical studies of glioblastoma, aging, Alzheimer's disease, and schizophrenia. And today he's going to talk to us about his long experience using different machine learning techniques to find imaging signatures of brain aging, early stages of neurodegenerative disease, as well as how to apply those methods to develop precision diagnostics for brain cancer. So please help me welcome Dr. Davatzikos. Thank you.
Thanks so much, Javier, for the introduction, and it's great to visit this beautiful campus and the nice group here at NIMH. So I got involved into machine learning back in the early 2000s, in the dark ages, where everybody was showing me the door when I was trying to convince them that machine learning is the way to go and multivariate analysis. At that time, I think there was one of the very first papers was a conference paper by Paulina Golan and others. I think Francisco and Tom Mitchell and others had one of the very first papers in brain decoding, and we had a paper in 2003 or so on classification of MRI scans of men versus women, older people versus younger people, fairly mundane things that you don't need an MRI scan to do that, but you have to start from the basics. Hopefully, we're doing some more interesting stuff now. So we had some ran some experiments with fMRI where we saw that you can look at the pattern of brain activity and tell whether someone is telling the truth or is lying. This was a mistake because there was a company that took this idea. They were claiming that they can catch cheating spouses or criminals or whatever. So I kind of got out of this quickly. But we continued primarily with structural brain MRI scans. And in 2005, in Archives of General Psychiatry, back then we showed, I think for the first time, that there's a fairly robust imaging signature, neuroanatomical imaging signature of schizophrenia that can be identified on an individual patient basis. And up to that point, there was there were a lot of studies looking at the group analysis and comparing patients versus controls, but it wasn't clear if you can actually see these neuroanatomical differences or peculiarities on an individual person basis. And after that, of course, there's been a whole bunch of things that machine learning has been useful for. Now the pendulum has swung the other way, and everybody wants to do machine learning, which is also a mistake. I think we have to balance somewhere in the middle. It's one of many tools that we have in our toolbox, and as long as we use it right and for the right task, I think it's very useful. So machine learning can do just quickly a lot of things. One of the areas that particularly deep learning has completely reshaped what we've been doing is image segmentation. This is one of the UNet, ResNet variations that one of the Jimmy Dorsey in my group has developed. It's replacing everything we were doing for years, for segmentation of brain tumors and skull stripping, to segmentation of small vessel ischemic disease, to segmentation of hippocampus and other brain structures. So now we're basically redoing our segmentation pipelines like everybody else, because I think for this type of problem, deep learning is great. But this is not what I'm primarily interested in, actually, because these are things that we can see, where the hippocampus is, what these lesions are. You have a tool that can do them systematically, reproducibly, and this is great, but it still doesn't augment our knowledge that much in terms of pointing the way to something that we don't see. But machine learning can actually tell us a lot about things that we don't see, but the machine sees. And in clinical neuroscience, if you look at the an MRI, a structural MRI of a patient with schizophrenia, I think every neuroradiologist will tell you that this is a perfectly normal MRI scan. It doesn't differ in any way from a typical MRI scan of that age range. But this person has a devastating disorder. Something must be different here that we don't see. It turns out we now know that computers can see that and can tell us what it is. The same thing with a mild cognitive impairment. This is an MRI of a healthy individual of about the same age, and this is basically a normal MRI for that age range. But this person has memory decline and might progress to Alzheimer's disease. We would like to know what's different here. And in oncology, the field of radiomics and radiogenomics has started to completely revolutionize things we can see in imaging. These are different channels that we have, the standard conventional images, T1 GAD, T1, T2 flare. This is time series from perfusion signals. This is derivatives of the diffusion tensor imaging and so forth. And it turns out, but with machine learning and writing the right feature, finding the right features in the right combination, we can actually tell a lot of things that we never expected, such as does the tumor have EGFR V3 mutation or not? And we now know that we can actually tell, and I'll show you some examples. Is the MCNT promoter methylated or not? Does it have an IDH1 mutation? What is the expected survival of this person, of this patient? How is the tumor infiltrating beyond the visible margins? All of these things are things that we can now tell with machine learning. And towards the end of my talk, I might have some time to show you a few slides on this. And along these lines, I think what I'm particularly excited about is that the machine learning can help us reduce the dimensionality and the complexity of multiparametric images and non-imaging data as well, in a way that can distill these complex multivariate patterns down to a fairly small number of informative biomarkers. And leading to dimensional neuroimaging. Of course, there is a number of dimensionality reduction methods. We will be using principal component analysis, independent component analysis, other things, non-negative matrix factorizations, and so forth. But they try to explain variance, and they reduce the data down to components that we cannot easily interpret. If you look at an ICA component or ICA components of a structural or functional signal, you can't really, you can't exactly interpret this reduction of variance, but machine learning can potentially reduce these signals in a different way that relates more to a clinical outcome, to a genomic variable, to a genetic variable, and so forth. For example, and I'll show you examples of subtypes of schizophrenia that we've identified in terms of brain structure, subtypes of mild cognitive impairment and Alzheimer's disease. So these can be different dimensions, and these have different prognosis potentially. Structural and functional covariance networks is another way to reduce the data. Patterns of amyloid deposition. So all of these dimensions can potentially summarize this complex multi-parametric data in a simple and fairly interpretable way. And I think we need that because the complexity of imaging and non-imaging data is becoming unmanageable. The other thing, and I'll come back to this busy slide later on, but machine learning can inform clinical trials in the sense that if you have personalized predictors of outcome, you can actually enroll into clinical trial people with a much more homogeneous profile rather than having heterogeneous populations which will need a large sample size to be able to show treatment effects. So a pattern is formed by sampling different parts of the brain, different images, whether that's a structural, PET image, functional MR, or other types of images. And you can, of course, couple it with demographics, genetic, clinical information, all these features that go into this black box that remains a black box to a large extent. And the black box will find out which combination of these and which of these features will give us the best prediction of the outcome variable that we're interested in. In much of our work, we've been calling this pair indices for spatial patterns for recognition of something, AD, schizophrenia, and so forth. This is some early work from Alzheimer's disease that shows areas, patterns that produce an index that is positive if you're expressing this neuroanatomical pattern, if you have this type of brain atrophy, and negative if you don't. Same thing for schizophrenia, and I'll show you more specifics on this. So it turns out that if you do that, whereas if you were to look at hippocampal volumes or total brain volume or other conventional measurements, you cannot really separate even Alzheimer's patients from controls very well. There are differences, there are significant group differences, but if you take an individual's hippocampal volume, you cannot tell if they have AD or not. It turns out that the SPEAR AD score can actually almost perfectly separate controls, healthy controls from patients. This is data from ADNI. And create an RSC that is almost perfect, very close basically to the accuracy of the clinical categorization into AD and control. And the areas that this pattern weighs more are, of course, hippocampus, temporal lobe, medial temporal lobe. You don't see some of the anterior stuff, posterior cingulate, perihippocampal white matter, some patterns of ventricular expansion, and so forth. All of this go together into a model that says you have this pattern or you don't have this pattern. But of course, you don't need an MRI to tell if someone has Alzheimer's disease. It would be too expensive and too uninformative. What you would like these indices to do is to predict future outcome. And this is, again, the SPEAR AD index looked at in patients with mild cognitive impairment. And these people convert to Alzheimer's disease at a rate of 15% or so per year. And so you would like to know in advance if you can predict which of the people with MCI are going to convert to Alzheimer's in the future. So just by looking at the SPEAR AD index, which tells you whether or not you have a pattern of atrophy that looks like that we've seen in Alzheimer's disease, just by looking at that, you can, and if you can split it, if you can divide it into quartiles, you can see that the people who don't have this pattern have much better prognosis than the people who have a full-blown pattern that we've seen in AD patients. And then in between. And if you add a couple of simple tests and ADAS-Cog, which is a fairly straightforward cognitive test that can be administered in the clinic, and ApoE genotype, which is taken from a blood test, you have very, very good prognosis of whether an MCI patient who shows up in the clinic is likely to remain stable for a long time, and this is a 5-year period here, of course, or progress very rapidly to Alzheimer's disease, or in between. What is even more exciting is if you can do that for cognitively normal individuals, and this is a paper we had in Brain back in 2009, where we showed from the Baltimore study of aging, this is a longitudinal study, that if you measure the presence of these patterns of brain atrophy, the AD-like patterns of atrophy in cognitively normal individuals, and if you look at the rate of change of them, you can predict whether or not they will convert within an 8-year period to MCI or will remain stable. Unfortunately, everybody had a positive rate of change, and none of us is getting younger. But the people who had rate of change, increasing prevalence of this AD-like atrophy, were almost distinguishable from those who didn't in terms of, and those are the people who progressed to MCI in an 8-year period. And again, you're talking about potentially 2 decades before these people become demented that you start to see these signatures, maybe 15 years, because even when they convert to MCI in this prospective study of fairly normal or supernormal, I would say, subjects, then they will take several years before they get demented. At least some of them. So you're looking at a window of opportunity for at least more than a decade, perhaps 2 decades before people get demented. And if there is an effective treatment, that's the time to administering it. So these very early markers can be detected. I think this is in contrast to what many people believe, that structural change comes last. That's, of course, if you're measuring brain volume and hippocampal volume, you're going to see it very late. But if you zoom into these more complex patterns, you can potentially detect them much earlier than other things. So back in 2005, we had a study in schizophrenia where, as I said earlier, we showed that you can detect a pattern, a frontotemporal pattern of I'm not going to call it atrophy because atrophy implies longitudinal change. It's of reduced cortical volumes in a number of areas. that gave pretty decent ROCs back then in a fairly small sample, about 150 people. So we have recently extended this into a consortium, and I'll show you results later on. But that was kind of the first indication that we can pick up these patterns of brain atrophy or reduced brain volumes in patients with schizophrenia on an individual patient basis. So machine learning has made a lot of progress, but there are a lot of unsolved challenges. And I actually wrote a commentary, an editorial, like 1 or 2 years back when I stepped down from the Neuroimaging editorial board about many of the challenges. But two of them are we're dealing with them pretty actively in the lab. One is, reproducibility and overtraining to a single data set. Unfortunately, a lot of the, especially older machine learning studies, were shown in single cohorts in maybe 100 or 200 individuals, and they are not very robust findings. Sometimes you have a lot of degrees of freedom. You can overtrain, you can overfit the data. You might be sensitive to picking up specific characteristics of the acquisition protocol that was used in this study versus that study. So reproducibility has been a main issue. The second issue is heterogeneity of biological patterns. If you're assuming that there's a single pattern of disease that you're looking at, you might be missing the fact that there are multiple, perhaps. And we know that there is disease heterogeneity in pretty much everything I've been working with, whether it's aging and cognitive impairment, schizophrenia, and other neuropsychiatric disorders, brain cancer. There's a lot of heterogeneity. So not one pattern fits all. So that raises the need for a lot of data from multiple sides, which brings up other problems such as harmonization and the need for advanced computational tools that do not require too much babysitting, because if you have 10 or 20,000 scans now, you cannot really be prepping your data too closely. You have to have fairly automated and robust computational tools that are feasible at that scale. So to address some of these, we've been leading the effort on a couple of consortia. One is the eye-staging imaging-based system for aging and neurodegenerative diseases, which tries to put together data from, I think, 13 studies. Right now, we have about more than 30,000 MRI scans. A subset of them have amyloid PET. A subset of them have resting state fMRI. The age range is from 45 and up. The majority is in the 60s and 70s. And actually, there's a fair number of data in younger ages as well. And some of them come from 1.5T, some of them come from 3T, and so forth. So there's challenges with harmonization. The second consortium is the Phenom consortium, where it evaluates heterogeneity in schizophrenia and psychosis more generally. And again, it involves groups all over the world, from London to Munich to Utrecht to Australia to the US to China. And we're trying to put together all this data. It's about 3,000 MRI scans, and some of them have longitudinal follow-ups, trying to better understand how reproducible and generalizable these imaging signatures of schizophrenia are, and tries to tease out heterogeneity. We had to solve the problem of harmonization, and we've been using a combination of COMBAT, which is a method that people in genomics have been using. It's a multivariate correction and harmonization method, along with GAMs using spline models to model nonlinear effects, such as age effects. So we've managed to put this effort has been led by Ray Pomponia, who is a research assistant in my lab, and who is also co-supervised by Hao-Chang Chu in biostatistics at Penn. And we managed to kind of obtain reasonable, at least seemingly reasonable, harmonized brain volumetric measurements of different parts of the brain. These are different studies having different colors, and you see that, and then the underlying model of aging trends is the one that is obtained by the spline GAM. So now that we put the data together, I apologize, I skip all the technical details, because otherwise I would give a lecture for each of these methods. Now that we put all the data together, we're able to observe a lot of interesting things that before it was kind of limited in small data sets. For example, this is the SPIRD score as a function of age in over 5,000 people. And you see that it's negative for the most part, which means that they're normal. They don't express this imaging phenotype or this brain atrophy phenotype. But after some age, the trend is in fact nonlinear or perhaps exponential upwards. And a lot of people start to develop this AD-like atrophy. And of course, that agrees with the frequency of the disease as a function of age. This is from a number of different studies here. And recently, the UK Biobank has been a major contributor, of course, to this. So now we put the data together. We want to start playing with this dimensional neuroimaging idea and create brain charts. Everybody likes brain charts. Whether people like brain charts, I'm not sure how useful they are, but they're fun to look at. The brain charts might be, I don't know, hopefully a little more useful and predictive, but for the most part, they're fun to look at. Right now, this work, Mohammed Habis has played a key role in this study. He's now an assistant professor in the University of Texas in San Antonio. So we started to create, of course, I cannot show you 20-dimensional graphs, brain charts, but I can plot 3 things at a time. So this is age, this is ischemic disease, white matter lesion volume, and the color here, or shown as isocontours, in this particular case is brain age, the SPARE BA, which is basically the MRI-based estimate of your brain age. You start seeing interesting things, such as for a 61-year-old who has relatively low load of these ischemic lesions, has a brain that looks like that of a 59-year-old, whereas if they have a lot of relatively larger volume of these ischemic lesions, their brain looks like a 69-year-old's brain. So you lose at least a decade or more if you have a relatively large number of volume of lesions. What is low and what is large there's no absolute value here. It's all relative to the population. So this is the entire population of 6,700 people, and you see that if you're an here, your brain doesn't look very good. This is memory now. I don't remember which memory score this is, but these are isocontours. So people, as you slide along this curve, you look at people who have the same memory performance, and you see that people who are a 60-year-old person who has high white matter lesion load cognitively looks like a 74-year-old who has a relatively low lesion load. So again, more than a decade difference. So these ischemic lesions are not a good thing, and at least they are the one thing that we can control, perhaps, by aggressive treatment of hypertension, diabetes, and all the other comorbidity, cardiovascular risk factors, basically. A finding that was perhaps the most provocative of all was that the presence of these white matter lesions doubles your probability of testing amyloid positive, even though presumably these are not directly related to each other. Potentially they are, or there's an underlying latent factor that drives both of them up. But these ischemic lesions double the probability of whether or not they're causal or whatever they are. They double your probability of testing positive for amyloid, which is quite important, I think. So now, I told you earlier about our 2005 study where we looked at the schizophrenia pattern. Now in the Phenome Consortium, we're able to look at almost 1,000 people, and we had a couple of papers. The first paper came out in 2017. There's another one in press now in Brain. And we found a very robust pattern of reduced brain volumes from prefrontal, orbitofrontal, some visual areas, perisylvian areas that had relatively reduced brain volumes. That in combination, all of this created a pattern that allowed us to build an ROC curve that is fairly respectable, of 0.83 or so. And we looked at it using leave one side out, do 10-fold cross-validation, and split samples and so forth. So it's a fairly robust imaging signature, even though this was across Germans, Chinese, and Americans. I was less optimistic in the beginning, but it turns out that there is a fairly reducible neuroanatomical imaging signature for schizophrenia. What is interesting is, we had reported in 2017, and Enigma and other studies have reported the same findings, effect sizes of reduced brain volumes. These green-blue areas are reduced brain volumes. Effect sizes that go up to the way to 0.5, and then some increases, which are larger ventricles, which has also been reported, of course, in the literature, amply in schizophrenia. But there was the internal capsule and the pallidum here were reported as significantly higher, particularly the pallidum. Both we and Enigma reported all this, but we didn't realize exactly where it comes from. You know, it was reported as part of the imaging signature. It turns out that it's very interesting, and I'll come back to this in a second. It relates to heterogeneity, but I would like to highlight these two findings right now, that 2 years later, we actually elucidated where they come from. Like everybody else, people have transitioning from conventional machine learning to some of these monstrous networks that I no longer know exactly what they do, but they fit the data, basically. It turns out that it doesn't quite matter what architecture you use. I'm exaggerating on this, but as long as you have enough degrees of freedom to fit the data and you make sure you don't overfit the data. But this is something that one of my students has been playing with in terms of looking at the brain age. The nice thing about this is that now you no longer need any pre-processing of the data, so you can estimate brain age from raw MRI scans without any processing, and you can apply it to an independent set. This is the UK Biobank, and you get fairly good predictions of age. Again, it's a mundane task, but the idea is that if your brain age shows that your brain is older or younger than expected, that says something about your brain health. The one thing that we found is that by pre-training, and a lot of you know use transfer learning or pre-training of these networks on brain MRI scans. So if we use the network that was trained to estimate brain age, and we try to now build classifiers of Alzheimer's disease, MCI, schizophrenia, and so forth, across the board, we found that the network pre-trained on and transferred from brain age to these problems does better than a vanilla, the same exact architecture trained on ImageNet, which is, of course, millions of natural images that people have typically used to train these networks. So pre-training on brain MRI scans with the mundane task of trying to predict the brain age helps build a network with weights that are more relevant to classification of AD, MCI, schizophrenia, and potentially other pathologies, especially if you have small sample sizes, which is all the things on the right that you see here. The blue is what we get with a pre-trained network. The green is what you get with the vanilla trained network. And you see that especially for small sample sizes, you start to lose your ability to build a classifier. So for smaller studies, it's important to have a pre-trained network. And this was just a fairly straightforward application. We're working on the idea of domain-specific networks that presumably capture the characteristics of brain scans and not of cats and dogs and tables and so forth.
You just send the MRs, or you also send the age when you start doing the classifying the Alzheimer's and the MCI?
No, that was sending the MRs.
Just the MRs. So even though you trained on the network with before that, obviously knew the age when it was trained.
Yeah. No, but these are initialized with the weights that you get from brain age, but they change afterwards in order to build a network that tries to differentiate between ADs and controls or schizophrenia and controls, right? So these three networks here end up having different weights because they each try to achieve a different task. It's just the starting point, which is important. Because these are highly nonlinear functions that you optimize. The starting point was that of the weights that we have found that predicted brain age. All right, so heterogeneity was the second major challenge, and I'll talk about that. A few years ago, I sent a couple of my students, we get a lot of bright engineering students, so I sent them off to study this problem of describing multiple or heterogeneous pathologies and kind of deviating from the assumption that we're looking for the pattern of schizophrenia, which is essentially a direction in the high-dimensional space that separates patients from controls. Of course, you have nonlinear classifiers, kernel machines, other things that, and now with deep learning, of course, that build highly nonlinear hypersurfaces that presumably separate patients from controls, but they are not very interpretable because you have a continuously varying pattern. So you cannot answer the question, what does the pattern of brain atrophy in schizophrenia patients look like? So we wanted to find the middle ground kind of solution where we had interpretable but nonlinear transformations between controls and patients. So Ayandong, who is now in industry, he approached this from a generative perspective. So this has a probability density function. These are controls, and these are patients. And we put all the patients in the same bucket without worrying at this point of their clinical phenotypes and potentially subtyping. We say these are all, and you try to be as broad as possible here, for example, including people with early-stage MCI, late-stage MCI, and AD altogether, and ask the question, how do these people differ from controls? So if this is the probability density function that controls have, you need a number of transformations to map it and to generate this probability density function of the patients. And then it turns out that there's a method that it's a generative method that solved this problem. A more discriminating analog is that of Erdem Barrels, who is now at Columbia University postdocing. He found a convex polytope that separates the control population from all the patients, and of course you need multiple hyperplanes to take care of multiple potentially disease subtypes or patterns, if you want. So, and of course, we tested this in various contexts of simulated atrophy and so forth. But then we started to apply them in different problems, and we found out that, as expected, of course, there's a tremendous heterogeneity, even in cases where we didn't expect as much heterogeneity as in the general, in a more general patient population. For example, this is data from ADNI. And we looked at we put together all the early, late-stage MCIs and ADs into the bucket of people with some pathology. And then we found that there are 4 patterns in which these people differ from controls. There was a 13% of these patients that didn't differ at all from controls. This might be because we were not able to pick them up, or because perhaps some of the controls were not exactly healthy, but they have pathologies, but they hadn't progressed yet to early-stage MCI or late-stage MCI. The 44% of the people had a more full-blown pattern of brain atrophy that included especially the medial temporal lobe and the hippocampus and some other areas throughout the cortex. This was what you expect from Alzheimer's patients who have a fairly pronounced atrophy in these areas. 15% of the people had very focal hippocampal and other temporal lobe atrophy, very focal. The rest of the brain was intact. And then the 28% had a generally global spread out pattern of atrophy that included the hippocampus. Otherwise, they wouldn't have been part of ADNI, but included a number of other areas. What was interesting is if you follow these 4 subgroups over time, and this is now in weeks, so it's a long time, of course. This is 6 years, 8 years, and so forth. You get that the people who had fairly normal anatomy, at least seemingly normal anatomy at first, had, in general, the best prognosis over time. It's the red group, number 1. Of course, as we know, this shows only data for late-stage MCI. If you are at late-stage MCI, chances that you will remain stable over time are low. But the people who had this pattern had the best chances of remaining stable. People who had the full-blown AD type of atrophy were the people who had the worst prognosis, the green here, which they converted fairly rapidly to Alzheimer's disease. People who had this focal hippocampal atrophy but pronounced were the people, the purple here, who initially had a good prognosis, but then they started to deviate and quickly quickly and rapidly started progressing to Alzheimer's disease. To the point that the ultimate outcome 8 years down the road in terms of survival curves was about the same as these people over here. So these are presumably the late-stage MCI patients who have early stage but progressive disease. And these people with a more generalized pattern of atrophy are the bluish, the number 3 here, the bluish line here, that initially they declined about 50% of them, but then they remained stable over many years, about half of them or 40% of them, which potentially means that at least half of these people did not have Alzheimer's or dominant. The dominant pathology was not Alzheimer's, but potentially something else, whether that was small vessel disease or other pathologies. So you see that even in a fairly homogeneous group of patients who were selected to have amnestic MCI and to be good candidates for having Alzheimer's disease, even in them there's heterogeneity, and this heterogeneity maps to longitudinal outcome. And I think some of these semi-supervised machine learning methods can tease this out. In schizophrenia, we had very interesting results, and this is a paper that is in press in Brain now. It was led by Ganesh Chan, who is in my group, and Gurai Eris and Dominic Doyer, who is in Munich. And we did a number of experiments that showed that repeatedly, reducibly, and robustly, there were 2 subtypes. This is the number of clusters that we got from the data. Now remember, we're not clustering patients. We're clustering differences between patients and controls. And this is very important because it makes it a little more robust or quite more robust to various confounds. Otherwise, you can start clustering data according to potentially demographic or other variables or genetic variables that might not be of relevance to schizophrenia. This shows the number of clusters that we got of subtypes, and this is we try different number of clusters. And for 2, we had the very reproducible ARIs, adjusted rand index, that was up to 0.7 and was highly significant compared to the null distribution that we got from permutation tests. So we looked at these 2 subtypes in more detail. We started to get very significant clusterings for higher number of clusters, but they tended to line up with sex and age and site, so we didn't really trust those results. At least at this point, we only had about 1,000 people in the analysis. So we focused on K = 2. We found basically that about 2/3 of the people, the ones who belonged to cluster 1, were the ones that had the typical pattern of brain atrophy that we expected and we knew of and that everybody knows of. A lot of orbitofrontal, prefrontal, temporal, perisylvian atrophy or reduced volumes that is present in this subgroup of patients compared to controls. These people also had seemingly progressive brain atrophy, and this is a cross-sectional study, so I shouldn't say progressive because it's not a longitudinal study, but it correlated with illness duration. But the 1/3 of the people that came out as a separate cluster had a perfectly normal brain, and they had increased striatum. So, and these are the people whose brain volumes didn't correlate with disease duration. So presumably, if we had longitudinal data, they might be more stable. We didn't find any other difference other than this between these 2, and other than the fact that the second subtype had was more educated, significantly more educated. But this striatum effect was not explained by medication. These results were robust to split sample, to also to cross-validation, to permutation tests, to everything else we did. They were consistent between men and women, so there has to be something there. And at the bottom is a difference between the 2 subtypes. But you see tremendous heterogeneity here. 1/3 of the patients are the ones who contributed to these 2 findings here, the internal capsule and the pallium that we had reported before. And we didn't realize at that point. But now we know that all these reduced cortical volumes comes from about 2/3 of the people, but 1/3 of the people contribute to these increases. I still don't know what the explanation is, and we try to. This paper is in Press in Brain. We try to publish it in JAMA Psychiatry, and people, of course, had this always raise the question, so what does it mean? But I think that it's important to understand that at the very least, we have to look at these people separately from these people. And now we're going back to the larger consortium with 3,000 people that includes first episode individuals, that has their, we'll look at richer phenotyping and hopefully genetics as well, to see what is different in these people compared to these people. But it's an example of, I think, very vividly shows you that we need to be very cautious about explaining group analysis, even if they're machine learning based, because ultimately under these umbrella diseases, there are subtypes that might be completely different. In certain areas like glioblastoma, for example, we now know that the glioblastoma might not be a single disease, but might be multiple diseases, depending on what mutational mix the cancer has. So I think we're moving rapidly towards precision diagnostics, which is important. I think I'll skip through this fairly quickly and say that there's unsupervised methods that we'll be using as well. This is my former student, Harini Ivani, who now leads a group in Microsoft, a research group. She applied decomposition, sparse, non-negative decompositions to capture covariations in resting state fMRI data and come up with a number of brain networks. Each of these components is a brain network or brain pattern, if you want. And we found very reproducible. This is essentially something that is analogous to ICA, independent component analysis, except that we've always thought that sparse non-negative decompositions are probably a more natural way to describe these functional covariations in the data for a couple of reasons. Number one, they allow overlap of these components, whereas independent component analysis, by virtue of looking at enforcing independence or looking for independence, doesn't like to have a lot of overlap between these components, even though the final results might have components that overlap. In principle, it doesn't like it because it violates the independence assumption. Whereas the sparse connectivity components allow you to have partial overlap, presumably because one brain region might mediate multiple networks and multiple functions. The second thing is that I found it difficult to, with unconstrained decompositions, I find it difficult to interpret the components, where with non-negativity constraints, you have only the addition of this component makes them more interpretable. And we have applied similar methods. Ari Sodras was also with me. We had a couple of papers in PNAS and Neuroimaging. He is now at Washington University as faculty, where we applied the orthogonally projective non-negative matrix factorization to both cortical thickness and regional brain volumetric data. And we found, I was kind of astonished by these results. It partialated the brain, basically. There was no a priori assumption here. We just said, look at the the gray matter volumes or regional cortical thickness, and look at how it co-varies across individuals, and try to decompose this covariation into a number of components using non-negative matrix factorization methods. And it ended up providing almost segmentations like, that of caudate, putamen, and thalamus went together into one component. The sensory motor strip is somewhere here. I don't see it in this plot. Supplementary motor cortex was there. There's the anterior and medial cingulate is here versus the posterior cingulate. The fusiform gyrus came out as a different region. So again, there's no other a priori assumption here. What it tells you is that the cortical thickness and regional volumetrics of these regions varies in such a systematic way across individuals that there probably is an underlying neurobiological reason why this happens, and that causes this coordinated variation of cortical thickness. But it's kind of interesting because we started to use this now as a data-driven parcellation rather than regions of interest that are derived from anatomical atlases that might or might not be the best way to partition the brain. So that leads to, again, I'm revisiting the dimensional neuroimaging slide, where now potentially these dimensions are not going to be principal or independent components, but are going to be dimensions of, are you expressing this pattern that we've seen in subtype 1 schizophrenia, or are you expressing this pattern? Are you expressing this or that pattern that we've seen in Alzheimer's disease? What are the patterns of amyloid deposition? We have similar results in there, and so forth. So these now, each of these dimensions has been informed by either clinical or genetic or other biologically interpretable variables that potentially can create a dimensional neuroimaging system that is both concise and interpretable. So finally, we've been doing this in neurooncology, looking at the glioblastomas, and again, this is another set of complex data. Our standard clinical protocol includes, the conventional images T1 gadolinium, T2 FLAIR, perfusion series, which capture the dynamics of absorption of the tracer, and which of course depend on the tumor. Derivatives from diffusion tensor imaging and so forth. There's spectroscopy data. There's models of biophysical models of tumor growth that we've been developing over the years. And it's very interesting because with machine learning, we can actually predict where the tumor is going to recur in the future. So glioblastoma has a very grim prognosis. It always comes back. But what they do right now is they resect the enhancing part of the tumor. This is baseline scans. This is what they would do right now. There's a peritumoral edematous region, which is both edema, and we all know that is also infiltrated tissue. But they don't resect this area because they don't know what is likely to recur. This is our predictions of what is likely to recur on this patient, and this is where recurrence appeared months later. This is another case over here where we predicted that this area here has been heavily infiltrated and it recurred early. So now there are a couple of clinical trials at Penn. One is delivering those elevated radiation, escalated radiation to these areas here, 75 gray instead of 60 gray that they usually deliver, to be able to potentially delay recurrence, at least in this area. And we've also been talking about potentially an extensive resection clinical trial that would try to go after these areas that show high probability of recurring. You cannot cut the whole area, of course, because you will kill the patient or create serious morbidity. But if you can go in a targeted and focal way, that would potentially change the outcome. We can predict patient survival as well, which is response to standard treatment, if you want, by using all sorts of different measures from imaging and demographics and even morphologic parameters of what is the size of enhancing tumor versus non-enhancing tumor and so forth versus edema. Putting it all together into machine learning, we showed back in 2015 that you can predict someone's survival. And of course, like I said, glioblastoma has a very grim prognosis. In 3 years, the majority of patients are dead, unfortunately, but some of them have very poor prognosis and some of them have better prognosis. And it's important to know who has good prognosis because someone who has good prognosis might go to a standard treatment and then be evaluated for pseudoprogression and then or true progression. But someone who has poor prognosis might not want to wait until the tumor comes back. They might want to be placed on immunotherapy or some other trial, some other therapy that might be more effective for them. And also our ability to detect treatment effects can go up dramatically, and this is a paper we have in the works now. We're trying to publish this. So say that you're trying to test a treatment, and the test treatment is red, so it prolongs patient survival, and this is a histogram of the patients who undergo standard of care. If you have 20 or 30 patients, you're unlikely to find significance of this treatment because you would need potentially 100 people or 200 people. If you had a perfect predictor of survival, so at baseline, if you know for each of these patients how long they're going to survive, you take the black curve and you squeeze it to almost a delta function. So this shows the difference between the actual survival and the predicted survival. Because you can predict everybody's survival, the predicted survival and the actual survival fall in line, and people who responded to the treatment fall below this line. So now all you need is a few patients to be able to detect treatment effects. More generally, the situation is going to be like this, but essentially what this says is that if I know up front how long the patient is going to survive, I can evaluate the treatment, whether the treatment worked or not, much better than if I don't. If I try to evaluate a treatment in glioblastoma where survival varies from 3 months to 3+ years, it's a very heterogeneous group. If I know that this patient was expected to survive, roughly speaking, 12 months, and they survived 15 months, then the treatment must have done something. So this is the idea here. So I think this machine learning has a lot of potential in this area in terms of informing clinical trials and treatments. So finally, this is from the field of radiogenomics, something that even to me was very surprising. We published this in Neurooncology a couple of years ago. We showed that by looking at different imaging features of the diffusion, perfusion signal, and T2 signal, and so forth, you can tell whether or not a patient has cancer with EGFR-B3 mutation, which is an important target for immunotherapies and other trials. This is our ROC that we got in the cross-validated discovery set and the replication set. It was a fairly robust signal, but small n. At that point, it was 135 people or so. So there were a bunch of imaging features, and I'm not going to go through the details. There were, the perfusion signal, the diffusion signal was different. There was these islands of pre-necrotic tissue that were features that characterized EGFR-positive tumors. But what was really striking that we didn't expect at all was this thing on the left here. This is the spatial distribution of the tumors in patients who did not have the mutation down here and who had the mutations up here. They appear this is the probability of finding a tumor in this location of the brain after deformable registration into an atlas. So these are almost disjoint distributions. So the location of the tumor relates to its molecular characteristics, at least in terms of the EGFR mutations here. I didn't expect that. And I don't know exactly why. Potentially the cells of origin, potentially the microenvironment, potentially higher migratory behavior. It's not clear why, especially since we don't have longitudinal data. But it's a very striking, I think, discovery of something that we didn't quite expect. Even though I've been working on the formal registration of brain tumors since 1997, it was more of an academic exercise for years. And now that we see the results, I understand that sometimes findings come out of where you don't expect them. For cancer imaging, we've tried to bundle a lot of our software into CAPTK, the Cancer Imaging Phenomics Toolkit, based on funding that we got from the ITCR, the Information Technology and Cancer Research Program here in NCI, and we're very glad to be able to do that. And I'll close my talk here saying that high-dimensionality medical images and multiparametric images hide a lot of patterns of that we cannot really appreciate, but they are there and they have a lot of information, and machine learning can tease them out. And finding such patterns is important for diagnostic and predictive purposes, understanding disease processes or patterns of functional activity. And machine learning can help us see things that we cannot see and quantify such patterns in a way that it can lead to a dimensional neuroimaging that is both concise and interpretable and informed by variables of clinical and biological interest. I'd like to thank a lot of the people who have now changed in my lab. Many of these people are going off to their own, form their own labs or to industry and so forth. And thank you for listening, and I'll take any questions that you have.